Third objective here is to discern between smooth muscle and cardiac muscle structure and function. And so we've been speaking a lot about muscle, but we've only been looking at the properties and features of skeletal muscle, which is one type of striated muscle. We're going to shift now and look at the other two types of muscle, smooth muscle, and then cardiac muscle. And we'll compare the differences again as we approach the last chapter, which has to do with the heart. So smooth and cardiac muscle, when we look at smooth muscle specifically, it has unique features that we don't see with cardiac or skeletal, because both cardiac and skeletal are both considered striated muscle in that they have this elongated, sort of striped or striated appearance because of the sarcomeres that are present on both of these types. So skeletal muscle has that, again, these sort of strand-like, fiber-like cells, and cardiac muscle has that as well. So smooth muscle is unique in that it doesn't have striations, and the muscle cells here are not organized into sarcomeres, which are these repeating units that we see on the other two striated types. Smooth muscle is also different in that it's typically found in our internal organs, so it's lining your hollow visceral organs like your bladder, your uterus, your reproductive tract, the gastrointestinal tract, and then blood vessels. So smooth muscle is typically lining most of our organs and blood vessels. It's going to be under our involuntary control, or I should say it's under involuntary control by the autonomic nervous system, which is the parasympathetic and sympathetic. And so we don't make decisions about heart rate, we don't make decisions about blood vessels movement or gastrointestinal tract activity. These are all involuntary control. Now the cells are spindle-shaped. These sickle-shaped or spindle-shaped cells that are adjoined to each other to form the entire muscle, again, not arranged in sarcomeres as we see with other muscles. And it's still going to have actin and myosin. So actin and myosin will be seen in all muscle types. However, the organization of actin and myosin is different here, okay? And then there are no sarcomeres, but there are dense bodies, and dense bodies are synonymous to the Z discs, which kind of anchor the cells together and stabilize them as the contraction happens. So we don't have Z disc or Z bands here, which are the boundaries of a sarcomere. Instead, we have similar structures called dense bodies. Now, the sliding filament mechanism still applies here, again, slightly different because we don't have a sarcomere, but actin and myosin are still being dragged against each other in order for the entire cell to shorten the way that we see in skeletal muscle. So the arrangement is different. It's more of an oblique arrangement where myosin, which is the thick filament, is in between two strands of actin, which are your thinner filaments. And the sliding here is more of an oblique sliding. So that causes the cell, which is this sort of oval-shaped cell, to scrunch down into a ball, right? And so that sliding model still happens, but it just creates a different shape. Instead of shortening in a horizontal way, it's scrunching this cell into a ball. We're also going to see the duration of contractions being longer here. And then myosin is going to slide across the entire length of the actin filament. So again, that's going to create a really sort of tight contraction or a really contracted or condensed state of the smooth muscle cell, as opposed to what we see with the shortening of typical sarcomere. Right? And again, the contractions here are much longer, much slower than the quick twitches that we see in skeletal muscle. Now, what are the steps that control the excitation-contraction coupling that is specific to smooth muscle? First off, the major difference is the source of calcium. Calcium in skeletal muscle comes from the SR, and it's released by way of the ryanodine and DHP receptor interaction and the T-tubules. In smooth muscle, we don't have all of those fancy mechanisms. So instead, calcium has to come from outside of the cell and as well as coming from the SR. So most of the calcium here comes from outside of the cell to drive this contraction. It's going to do so by way of voltage-gated calcium channels that are on the cell membrane or the sarcolemma. And then when calcium comes in from the extracellular fluid, it actually triggers more calcium to be released from the SR. So most of that comes from outside, but then it triggers what's known as calcium-induced calcium release, which further triggers more calcium from the SR inside of the cell. Calcium will then bind to calmodulin. So opposed to what we see in skeletal muscle where it binds to troponin, here it binds to another regulatory unit or protein called calmodulin. And then together, the calcium calmodulin complex will activate another protein, myosin light chain kinase, another enzyme rather, that is actually going to phosphorylate the myosin head, and that will drive the myosin dragging and drive the cross-bridge cycling. So excitation, contraction, coupling, and cross-bridge cycling and the overall sliding filament model happens in smooth muscle, but it just looks different in terms of the players and the structure of the cell. So here's a schematic that just goes over the steps here. So calcium coming from the extracellular fluid, mainly, when it comes into the cell, it triggers more calcium release from the SR. So calcium-induced calcium release. From there, it's going to bind to calmodulin, and it's going to make the calcium calmodulin complex. So once this complex is formed, that can actually go and activate the enzyme MLCK, or myosin light chain kinase. Now, this enzyme, once it's activated, it's a kinase, so it's going to phosphorylate or add a phosphate to myosin, the myosin head, and that is what will drive contractions. So if we have no MLCK activated because we have no calcium and therefore no calcium calmodulin complex, then we have the unphosphorylated state of the myosin head, which means there's no cross-bridge activity or no contraction. This state allows the muscle to relax. And then on the opposite end, when we have calcium present and we have MLCK activated, and now MLCK can phosphorylate the myosin head, now we can see cross-bridge cycling and therefore contraction. So notice that calcium is still playing the regulatory role here. It's turning things on and off, but just in a different way. It's binding to this complex, and the complex in turn activates the kinase that will phosphorylate the myosin head and turn contractions off, or dephosphorylate the myosin head and relax the muscle and turn contractions off. Okay. And this is just the schematic that I drew out, same events that are happening, but I'll share this just in case you want to practice going over the events here. Really important to think about how this complex is formed only when calcium is present. So calmodulin is floating around in these smooth muscle cells, but it's not activated until calcium binds to it. And then it can activate the enzyme, myosin light chain kinase, and then we can have phosphorylation, cross-bridge cycling, and contraction. All right. Now, how does the muscle relax? So smooth muscle relaxation is dependent upon the removal of calcium, very similar to what we saw with skeletal muscle. So calcium ATPase is going to pump the calcium back into the SR, but because some of that calcium also came from the extracellular fluid, we now have calcium sodium countertransporters. Remember, countertransporters are going to move calcium in the opposite direction by using the energy harnessed from moving sodium in its natural passive direction. So that is going to be another mechanism for getting rid of some of the calcium from the cell. And so that is how we can unphosphorylate the myosin head, and then the muscle can relax. Now, what we discussed earlier is the contractions here are slower, 10 to 100 times slower than we see in skeletal muscle. So skeletal muscle contractions are very short and very quick. Even the longer end of that scale, which is our slower contracting muscles like our postural muscles, even they contract faster than smooth muscle cells do. So the contractions here are much more sustained, much slower to start, but also to go back to relaxation. And in terms of the regulation here, the nervous system plays a huge role, especially the autonomic division, the parasympathetic, which slows things down, and the sympathetic, which usually speeds things up. So this can be excitatory or inhibitory depending on the exact receptor. So looking at all the receptors we discussed, the adrenergic receptors, the muscarinic receptors, depending on what's binding and whether they're activated or not, we can see excitation or inhibition at those junctions. The responses here are going to depend on the neurotransmitter that's released from the varicosities as well, and then how that neurotransmitter crosses the neuroeffector junction, binds to its corresponding receptor, and then the signaling cascade, right, the second messenger cascade that will bring about a specific response.